Hi, I'm Lauren Mullen, Principal Scientist in the Scientific Operations Group here at Waters Corporation. In this segment, I'll discuss how ion mobility and collision cross-section, or CCS values, combined with data-independent acquisition, or DIA, can help improve the discovery metabolite identification process. These innovative technologies can help make faster and better decisions in the design, make, test, analyze cycle for drug design and medicinal chemists. I'll go over what is traditionally done for metabolite ID in discovery, review the concepts of ion mobility, collision cross-section, and DIA, and see how they can currently provide better data in the drug discovery phase and what future uses there might be. All of the data I'll show was acquired using the Vion IMS QTOF, which integrates ion mobility and CCS values in a routine, accessible manner. Discovery is less focused on detailed information for each candidate compound and more focused on narrowing down a large list of potential candidates to those most likely to succeed. At this stage, quick and high-level evaluations of compounds and their structures are necessary. For example, drug discovery teams need to manage clearance so that compounds have an acceptable half-life. Metabolic routes, which are of toxicological concern, need to be identified, and any potential for monozymic metabolism highlighted. And that's all within the context of the classical drug metabolism challenges of complex matrices, shifting retention times, analyzing cross-samples, and the need to be as specific as possible about sites of metabolism, rather than just reporting on oxidation. Unless DMPK labs can provide feedback on the metabolic fate of compounds within a few days, medicinal chemistry teams will already have made decisions about their next set of targets, without the benefit of knowledge of metabolic liabilities which could be designed out. Another limitation is the use of traditional data-dependent acquisition, or DDA methods, to obtain MS data. DDA methods typically involve generating an inclusion list of likely metabolites a priori so that as many target metabolites as possible can be identified. These methods must, however, be customized and are time-consuming to set up, requiring generation of customized lists that are typically different for different samples and/or repeat injections of the same sample. So how can this process be improved? Are there technologies available that are robust and generic enough to speed up this workflow, while at the same time giving clean spectra for discovery scientists? The answer is yes. Ion mobility separation, IMS, combined with data-independent acquisition, DIA methods. DIA methods collect all mass spectra within a wide mass range. This reduces the potential for mismetabolites, and the data can be reinterrogated in the future. But this can also be time-consuming because of the amount of data that is produced. MS to the E rapidly alternates between full-scan, low collision, and high collision energies, allowing both precursor, drug and metabolite, and product ion, high-resolution, accurate mass measurements to be obtained. To help solve this challenge, ion mobility was introduced as a way to filter through the data and take advantage of DIA techniques. Ion mobility provides an added separation dimension based on an ion's size, shape, and structural conformation as it passes through a drift gas. When combined with MS to the E, this acquisition method generates high-definition MS to the E, or HD MS to the E data. So what is a CCS value? Collision cross-section is a physical-chemical property that can be determined by measuring the ion mobility of a molecule. This value is related to its chemical structure, mass and size, and its three-dimensional conformation, or shape. Here, CCS is represented by the molecule trapped within a sphere. Its CCS is the shadow cast by the sphere. How do we obtain a CCS value? Following ionization, molecules travel through a nitrogen gas-filled drift cell. A direct current is applied here, creating a waveform which the ions surf. Based on the molecular size, shape, charge, and charge distribution, ions will travel at different rates through the mobility cell. This time to travel is referred to as the drift time, measured in milliseconds. All calibrations and calculations are performed automatically by the software, making integration of this powerful technique possible. CCS values are especially valuable when using a DIA-only approach to analyze samples, which collects all of the data all of the time. The resulting MS-MS spectra can be mixed, leading to incorrect metabolite identification. When applying IMS as a means of improving selectivity, the MS-MS information is matched to the appropriate metabolite precursor mass using CCS values, thereby improving spectral clarity, aiding in identification, while still gaining the benefits of a DIA workflow. Improved spectral clarity means an increase in the number of identified fragment ions over the number of false positives for structure elucidation metabolic fate. This leads to better informed decision-making regarding drug design ideation. Experimental CCS values, together with CCS predictions, can also be used to localize and reduce the number of possibilities for isobaric metabolites. This is an exciting topic of interest with implications for future uses of CCS. More recently, interest has grown to predict CCS values using machine learning-based algorithms for compounds for which no experimental reference or library data exists. This type of information can be used not only to differentiate between isobaric compounds, but possibly also aid with the site location assignment of structural isomers. CCS value measurement has the potential to aid in drug design and drug metabolism studies by confirming the CCS values of plausible metabolites, or at least greatly reducing the number of candidate structures. Ultimately, the use of ion mobility, CCS values, and DIA in drug discovery gives metabolite ID scientists better quality and more robust data, allowing them to make better decisions faster. The VION provides a data acquisition platform that makes this process accessible and routine. We hope this overview of the benefits of IMS, CCS, and DIA has been helpful. Thank you very much for your time today, and for more information on this topic and others, please visit the link below.